people ask me then, well, Mike, if it's fundamentally over, why are we still getting hundreds of deaths a day from SARS CoV-2? And I've thought a lot about this. There is a test that's performed. People have their noses and tonsils swabbed, and then a test called a PCR test is performed on that. And what they're looking for isn't the virus. You might think it's looking for the virus, but it's not. What they're looking for is a small piece of genetic sequence. It's called RNA. Unfortunately, that bit of RNA will be found in people's tonsils and nose, not if they've just caught the virus and they're about to get ill or they're already ill. It's also going to be found if they were infected previously, weeks or even sometimes a small number of months ago. Let me just explain why that is. If you've been infected and you've fought off the virus, which most people do, you'll have broken, dead bits of virus. These are tiny things, smaller than your cells, perhaps spread all the way through your airway, embedded in bits of mucus, maybe inside an airway lining cell. And so over a period of weeks or months, you bring up cells that contain broken, dead pieces of the virus that you have conquered and killed. However, the PCR test is not able to detect whether the viral RNA has come from a living virus or a dead one, as I've just described, the virus. Why are we using this test that cannot distinguish between active infection and people who've conquered the virus? This test has never been used in this way, and I've worked in this field. It's not a suitable technique. It's the kind of technique you would use for forensic purposes. If you were trying to do a DNA test to establish whether or not a person was at the scene of a crime, you would not be doing these tests by a windy supermarket car park in what looks like plastic marquee tents on picnic tables. It's not suitable at all, and it definitely shouldn't be done in the way it's being done. It's subject to many mechanical errors, shall we say, handling errors.
And that brings us to how we're interpreting test results wrong. Think of those two samples, one full of virus, the other with only trace amounts. In either case, the results will be returned as positive. But if you remember, the power in testing is not just knowing you're sick, but in identifying people who are infectious before they can infect others. Generally, the approach has been that if someone tests positive, then we interpret that they are actively infected and are infectious. The problem is both of these interpretations are not necessarily correct. First, testing positive doesn't mean you're still infectious. To be infectious, which means being able to make someone else sick, requires two things. First, it requires that you are shedding virus. Shedding virus means that virus is found in your droplets created when you sneeze, cough, or breathe. PCR testing absolutely tells us whether you are shedding virus. The second thing infectiousness requires, though, is that the virus you're shedding is viable. In other words, that the virus can survive and grow. PCR doesn't do this. To do that, we have to isolate the virus sample in the lab, culture it, and see whether it replicates. So PCR doesn't actually measure infectiousness.
The cycle is typically repeated 25 to 40 times, and it is important to realize the exponential amplification of material that occurs with these numbers. For example, at 40 cycles, there would be around 1,000 billion copies produced. However, Taq DNA polymerase has a certain number of mismatch errors, so higher cycle numbers are more likely to produce inaccurate results. It is also important to realize that a typical PCR test only detects a small sequence of genetic material. It does not detect anything close to the entire genome of a purported organism. With a European court declaring the tests not fit for purpose and potential lawsuits brewing, last month the WHO quietly issued a statement suggesting more caution with so-called positive tests.
I want to ask this to Kary. How do they misuse PCR to estimate all these supposed free viral RNAs that may or may not be there?
I think misuse PCR is not quite. I don't think you can misuse PCR. No, the results, the interpretation of it. See, if you can say, if they could find this virus in you at all. And with PCR, if you do it well, you can find almost anything in anybody. It starts making you believe in the sort of Buddhist notion that everything is contained in everything else, right? I mean, because if you can amplify one single molecule up to something that you can really measure, which PCR can do, then there's just very few molecules that you don't have at least one single one of them in your body, okay? So that could be thought of as a misuse of it, just to claim that it's meaningful. PCR is separate from that. It's just a process that's used to make a whole lot of something out of something. That's what
Also,
it is. But it's not, it doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that. It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that.